It's a pleasure to introduce Terry Hazen. Terry is a microbial ecologist. He received his PhD in Wake Forest in microbial ecology. He was a professor in microbiology at the University of Puerto Rico for many years. He then moved to several DOE facilities, eventually coming to the Berkeley Lab. He's a member of the Earth Science Division, has a leadership position there, his leadership position at EBI, leadership position at JBay. He's a leader. He's been, he's a microbial ecologist who's had a real focus on bioremediation. Has more than 200 manuscripts in this area, but I think importantly, he's really had an impact. He has several patents as well as R&D 100 awards that are being applied, in that he's that there, he both writes about bioremediation, and his research has led to impact on bioremediation. And so it's a real pleasure to have him. He's going to talk about the Gulf oil spill, ecogenomics, and ecoresilience.
Okay, so to more or less pure biology, and from what we have now, we also have a very large team. And what I would like to say first, for full disclosure, that we started out with funding from the Energy Biosciences Institute, which 4 years ago won a BP grant for $500 million for 10 years, and to establish the Energy Biosciences Institute, and the lead organization is the University of California at Berkeley. We have a program that's been going on for about 2 years in microbial enhanced hydrocarbon or oil recovery. So we were working on these types of issues with ecogenomics and oil, and actually samples from the Gulf 2 years ago, and actually had all of our techniques and everything else. So when this happened, we basically raised our hand and said, would you like us to help? And they said, come on down. And so we did. And so we got in there quite early, and as you'll see, we were out in the field for a long period of time. I'm certainly speaking for the entire team here, and our first paper in Science had over 31 co-authors. And I always felt like I was missing somebody. Today even, we have a large number of other people that have been involved, including a lot of Eddie's folks and at JGI. And in particular today, the two gals on the end here, Janet Jansen and Olivia Mason, helped me prepare some of the slides that you're going to see today. What happened? So on April 20th, the Deepwater Horizon drill rig sank, and what happened is the Deepwater Horizon drill rig, which is about 50 miles off the coast, exploded and sank for unknown reasons. It burned for about 2 days. 11 people lost their lives. And when this is working, and you can get it from this website, you could actually see it collapse on the bottom. It was about 1,500 feet deep, and it was a light crude, and they initially started with ROVs, and there was a lot of oil coming along the surface, up to the surface. Their initial estimates were 5,000 barrels a day, but basically, that was proved to be drastically wrong, and the actual amount was more than 60,000 barrels a day. It went on for 84 days. They tried various strategies of containment caps, top kills, and because all of the emergency backup systems failed, and the blowout prevention device failed. This was one of the deepest wells that's ever leaked at 1,500 meters. and they had to use ROVs, remote operating vehicles, to do a lot of the work. And of course, BP and the oil companies were the only ones that had those. So this is a very light crude, and you can see here in the top the actual composition of this crude. And I brought some of it with me. And since the well is now dead, this is more valuable than platinum. And you can see how light it is. And in fact, 40% of it will volatilize within just 2 weeks, and more than 30% within just 2 days. As opposed to weathered oil, which is the same Macondo oil, you can come up and look at this later, it looks more like motor oil. So once it's gotten weathered and it goes to shore, it looks completely different, and it biodegrades completely differently. Also, I have some oil and dispersant. There was a lot of talk about dispersant here. So you can see how that actually looks. And you can see a little oil with the seawater and see how it dissolves into small droplets. And that's actually one of the things we'll talk about, because what we found was that the oil was being jetted into the subsurface, into the deep sea at 1,500 meters here. And let's see if I can get my, boy, this does not want to cooperate. You can see here at down deep that because it's being jetted, it's a lot of droplets are forming. Now, if the droplets are 10 to 60 microns, they're neutrally buoyant, and so they moved out with this current between 900 and 1,300 meters and formed a very large plume about the size of Manhattan. But this plume was very low concentrations. Highest concentration was 10 parts per million. Average concentration was a little bit higher than 1 part per million. So quite low concentration, and that escapes a lot of people. And of course, there was a lot of natural seeps, and we'll talk more about those later. They also, of course, were jetting in, trying to cap this, putting in lots of drilling mud and that sort of thing. There was some oil with the drilling mud that went around the blowout prevention device, but we basically did not see much oil at all and haven't been able to detect any farther out in the sediment. The rest of the material went to the surface, the larger droplets, and there it floated to the surface, and of course they had skimming, dispersant. This was so volatile and so flammable that they in fact could burn it directly. They also used dispersant, and the dispersant this was the deepest application of dispersant that's ever been done subsea. It's corrects at 9,500, and basically they were doing it for safety reasons, because they felt that if the oil was coming directly to the surface, that it was going to be so flammable that they were going to have to abandon leak operations or well control operations here. Within 4 hours of adding the Crexit at the wellhead or at the riser, when the oil was coming out of the riser, it basically moved far enough away that it gave them a safe operating distance. They also applied dispersant above, and of course you got a lot of weathering, a lot of evaporation, and things like that going on too. And of course they also removed some of it, captured it through the pipe that they eventually got on there. 4.9 million gallons. It's the second largest marine spill in the world, the first being the Gulf War. Until Deepwater Horizon, there was another spill that was second. It was also in the Gulf of Mexico, and it's the Ixtoc 1 blowout. And we'll talk a little bit more about that. But you can see the relative proportions here that were dispersed, naturally dispersed, chemically dispersed, and then of course captured and skimmed, and 31,800 people were involved with this. At one point there was over 7,000 ships involved, They cornered the market on boom, and in fact, there was at one time, there was no more boom available anywhere in the world. I'm not sure that was the best idea, but we'll talk more about that later. And of course, 440 controlled burns, and they applied 1.84 million gallons of dispersant. So what we wanted to do was apply a sort of oil spill systems biology approach, looking at exactly what happens at all levels, from the ecosystem right down to the molecular level, going through a series of steps with communities and populations, and then potentially developing models for cellular properties, communities, and then finally ecosystems. To do this, we used a number of approaches that we have at our expo at the lab here that we've developed. And this includes microbial community and structure and function analysis using viral chips and geochips, and then metagenomes and metatranscriptomes with JGI, and then also actually isolating and taking bacteria and determining rates of biodegradation, and using AOS, a variety of different techniques that we've developed in the lab, including phenotypic microarrays and things like this. And then also geochemistry and isotope analysis, and basically looking at trace amounts of daughter products and contaminants that were there, and critical nutrients. You can see here the systems biology approach, basically sort of labeling a little bit more clearly all the different techniques that we simultaneously applied, looking at this in a complete systems biology approach. We were on 4 different ships from May 25th until October 20th, and this is totally unlike a lot of the other researchers that were out there, which only had missions of 1 to 2 weeks at a time. We were out there continuously, and I was constantly moving people back and forth. So it was, I got a lot of calls late at night, and of people that got tired of being out there and having to replace them, etc. We also had a couple of people that went, how should we say, native on me. They started smoking and playing cards with the deckhands. And so we had to retrain them when they got back. But they got to stay down there for a long time. That was good. So we fixed samples for ecogenomics, filtered them, and then fixed them with -80, frozen, flash frozen, right on board the ship. We had -80s right on board the ship. We also did a variety of chemical analyses and fixed material for isotopes and various types of nutrient analyses, and of course also took live samples back at 4 degrees C. We went from 1 kilometer, or as close as we could get to the wellhead, out to more than 300 kilometers in all directions. We had collaborators at University of Alabama, Florida State University, Gulf Coast Research Lab, Rutgers University, University of Oklahoma, Penn State University, University of Georgia, Scripps, and the University of Tulsa. If you go to this page, it's been up from day 1. There was never any restrictions on any of our data, and so instantly when we collected data and we had daily reports, it was put up on that web page. And you can see all that, and it's been a great outreach thing for folks. Here you can see they're flaring gas after they were capturing some of the oil from the riser, and this was going on all the time. As a matter of fact, so much gas was being flared that they had 2 or 3 ships that were constantly dousing it with water. They also, here you can see the controlled burns. These were big burns. Quite often we would see mostly this type of an oil sheen on the water. But occasionally we would see these droplets and stuff, and occasionally we would see this typical type of light crude mousse, which is bright red, and it's typical of light crudes. We also, whenever the ships came inshore to ship samples and get resupplied, they would be down for a day or so, and I'd send the folks on board the ship out to get samples of oil that was coming ashore, so we have those samples too. We took samples with a variety of devices, mostly with the CTD, and that's what I'm going to concentrate on today. These are conductivity, temperature, depth devices that can be lowered in the water. They have a carousel of 20-liter bottles that can be fired independently. And then we had, for surface oil, we had the Halliburton bucket, and that seemed appropriate somehow. And you can see here we were filtering in the lab. We had a complete lab set up on board the ship. So here they are, close to the wellhead, actually taking water samples from the CTD. And what we had on board that CTD was two fluorescent devices, so we could see the oil in terms of fluorescence, and they were calibrated to different wavelengths, but wavelengths that would have picked up the oil. So we would go completely down to the bottom with the CTD, and then on the way back up, we would take a sample below, within, and above the plume that we were detecting, and then at the surface too. We also had a lot of other measurements in terms of dissolved oxygen and pressure and nitrogen and a variety of other things that were on board those ships. Now, 2 days before we published our paper in Science, Woods Hole published theirs, but they were looking only for the deep plume. They were using a number of different devices to actually drag back and forth across the plume to define it. They did an excellent job of that. And so that was Dr. Camilli et al. And they found it exactly where we found it, and of course beat us to it by a couple of days in Science Express. This shows the samples that we took. And the plume was going off to the southwest because that's the direction the current was flowing. Now that was a good thing because the dead zones were up here to the northwest rather, and of course the coast is up in this direction. What did we find in terms of oil down deep? Well, most of it was alkanes, and the total alkanes you can see in this particular strata, and of course it went out as far as 50 kilometers. We can also see BTEX compounds, so these are benzene, toluene, ethylbenzene, and xylene. The PAHs, which were low in concentration and also quite light, we only found those at the surface. So what did we see? Well, what happened was we see this at 1 kilometer. We see this intense amount of oil in terms of the signal, but again, it's only, at most, it was 10 parts per million. And we see the bacteria densities were about the same, these dots here. But out at 5 kilometers, the bacteria densities have doubled. The oil has gone down, and then out at 10 kilometers, the oil has nearly disappeared, and bacteria are still quite high. But at control sites that were not contaminated in any way that we could discern, and we're far away from the well, you don't see any of that change in density or bacterial density or in that fluorescence. We also saw dissolved oxygen dips. So a very slight dip here at 1 kilometer, but a major dip at 5 kilometers, and less so at 10 kilometers. But of course, a lot of the oil had been degraded by that time. Statistically, we could show that, in fact, there were decreases in phosphate in the plume versus non-plume. Now, I'll call this plume versus non-plume, but really a better description is cloud, because it was such a low concentration, very small droplets, and actually, except in one sample, you could not see it or smell it in the water samples that we were taking. We also saw some changes in ammonia that went up, and of course, then nitrate that went down. Of course, all of these things indicate biological activity and biodegradation was going on. Of course, all of the hydrocarbon components were significantly higher in the plume. We also saw a major change in the diversity and species richness. So species richness and that sort of thing went down. Not with archaea, because in fact it was an aerobic environment and there was no anaerobes down there, relatively. And we also saw major changes in a lot of these other parameters, which also indicate, again, the Oceanospirillum strain that we'll talk about some more here in a minute, and also lipids, total lipids, and some lipid parameters that we know are associated with oil degradation. In looking at the lipids, you can just see a major change in the biomass between the plume and non-plume. Also, you can also see from these signature compounds major changes in both the plume and non-plume sites. So we initially used our phylochip, which has 50,000 different taxa on it. It's a highly redundant system. There's nearly 2 million probes on that chip, and we can do these tests in as little as 8 or 9 hours. And so we could get an immediate relative density of particular species. We found 962 subfamilies in 62 different bacterial phyla. And however, only 16 of those subfamilies in the gamma proteobacteria were significantly elevated in the plume. So quite a bit of difference. We could also show that, in fact, all of these subfamilies that were found to be elevated in the plume, in fact, have members that have been known to be associated with oil or with oil biodegradation. When we looked at the lipids versus the 16S rRNA with the phylochip, we found amazing similarities. The plume and all the samples that we took from within the plume versus non-plume, and here with the phospholipids, plume and non-plume. So they agreed quite well, and in fact we've seen that consistently. You need multiple lines of evidence whenever you're doing something like this that has the public's attention. And we applied all of the techniques that we possibly could to this. So we did clone libraries, and we did about over 500. And you can see here from non-plume sites, we were getting this particular Oceanospirillum clone in only about 5% of the clones. But either 95% or 90% of the clones from the 2 sites that we looked at and did clone libraries from within the plume. We also did pyrotag sequencing, and here you can see the rarefaction curve, and you can see the uncontaminated samples that were about 37 kilometers away versus the proximal part of the plume and the distal part of the plume. So it even became, as the plume moved farther out, it even became fewer number of sequences and less species richness. You could also see this when we do this pyrotag comparison. And using some of the new techniques at JGI, we did single cell sequencing, or I should say they did. And we targeted Oceanospirillum and Coeliaceae, and you can see large percentage of those, over 90%, in the 2 plume samples were the ocean aspergilles gamma proteobacteria, but dominant of the gamma proteobacteria. And then we also saw some of those in the uncontaminated samples, but the diversity was much, much, much greater. Now, it's interesting, we did a raw metagenome blast and looking at the community composition, and actually got something quite similar. So it was, even though it's different, but even without doing any stripping and manipulation and a variety of other things, in terms of the sequencing here, we still got basically the same results with these gamma proteobacteria being dominant in these plume samples. Now, we have known for decades that every component that is in oil, bacteria can degrade. So it wasn't really surprising that they had this ability, but we applied the University of Oklahoma GeoChip that Joe Zhao's group has developed, and this is a functional gene array. And you can see here the number of different categories that he has on this, and we could then tell the amount of expression that was occurring in these particular genes that might be involved with oil degradation. So what we found was these alkane degrading genes were all more expressed in the plume, as were all of these aromatic degrading genes. And you see almost all of them are significantly higher. In addition to that, we correlated them directly with that amount of expression with the amount of oil that was in that particular sample, and found what particular components in the oil, they were highly correlated. So there was more gene expression for those particular components in the oil that we know that these particular genes could degrade, and these particular proteins could degrade. So again, further proof that these things were happening. Now, in a single cell genomics, way too much information here, but this shows the metagenome and shows the match that we got with the single cell genomics. And just to blow that up a little bit, you can see that better here. And you can see the ocean aspergilles. I won't show you the Coelliaceae, but this is just an example of that. So from that single cell, we've developed a model, and we're still working on this, but I believe you can see here that it has flagella, and these were highly expressed. It also has a methyl-accepting protein for chemotaxis. That was highly expressed. So these bugs were oil-seeking missiles. And in fact, that we'll explain in a minute why they responded so fast. They also had a lot of ability to transfer different limiting nutrients across the membrane. And they had a plasmid. We're still working on that. They also had a lot of ability to degrade different alkanes in the C6 to C20 range. This is the actual ocean aspergilles bug, and I probably underestimated, so I do all these counts myself, actually, and I like to see what's down there. And so at 1 kilometer, you can see that this bug has actually started to divide. If you can see this well enough here, I'm not sure you can see this well enough in this room, but there's actually little divisions here. And so they've already started to divide. Now, at the rate that that current is moving, it would only take a matter of 7 or 8 hours to get out that far. So they've already started to divide and started to multiply. When they get out to 10 kilometers, they've completely divided. They're fat and happy, and there's lots of bugs there. Now, we had a problem. Remember that the temperature at this depth, maximum density of seawater, is 4 to 5 degrees C. And it stays that way all the time at this depth. So these were psychrophiles. They actually grow better and degrade oil faster at 4 degrees than they do at 20 degrees. So when we took live samples back, surprise, surprise, all we found was flocks, no oil, because the bugs were there and they were doing fine at 4 degrees. And so we went to the synchrotron FTIR, and Ho-Ying Holman, in her setup that we have at the Advanced Light Source, was able to show that these flocks have the bacteria associated with them, and these are bacteria, proteins, and carbohydrates. There was a very small amount of alkanes that could be detected in those flocks. And then we had oxidation products, nitration products, and sulfation products, so they're very active against, for degrading all of this oil. And so the best way to describe this is bacterial poop, probably. And in fact, that's all we had left. Now, when we brought stuff back from the surface, it was a little bit different at 4 degrees, because the surface water temperature is, geez, sometimes it was 30 degrees C out there. Big droplet of oil. So you can see the alkane heat map here. And then you can see, there's a bacteria attached. And what do you know, if we look at different oxidation products from the IR spectra, we can see that there's a lot of activity associated with that bacteria that's attached to that oil droplet. And here you can actually see the bacteria attached to that oil droplet and how they were actively going after it. Now, how fast could these bacteria degrade this? So we did 4 different techniques. We used 2 sets of data from ourselves and from data, I call it the BP data. Technically, it was Lancaster Laboratories and Louisiana State University did the analysis, and they did not send me anything that was analyzed previously. I got the raw data. People always ask that. So I got the raw data. We analyzed it and calculated on a conservative estimate of how fast it would take to go out 10 kilometers, then how fast the alkanes were degrading, because the alkanes were the dominant thing that we were seeing in the oil down deep. We also did a standard technique of using media, minimal salts marine media with peptone to grow up the bugs to higher densities. And we did a couple of passes with that, then took out the peptone with that marine media and added oil. This is the standard way they do oil degradation studies. However, I didn't like that, so we also used a microcosm. So this is water that was not within the plume. It was not contaminated, but it was from the same depth. We incubated it at 5 degrees and then added fresh Macondo oil to it and calculated the degradation rates at these different points. Now these are half-lives. So of course, if I say the half-life is 1.2 days, it doesn't mean in 2.4 days it's all gone. It means half of it's gone in 1.2 days, and half the remaining is gone in 1.2 days. And so it does take a long time for some of those components to degrade. But how fast does it degrade? 1.2 to 6.1 days. So people were amazed by that, but actually we weren't. In fact, we also showed that the alkane numbers in terms of C26, C27 to various other lower numbers of alkanes changed dramatically as we got farther and farther away, which it should do if they were being degraded. If we go to the literature, we see, in fact, our data for 2 different types of techniques, microcosms and what was in the oil as it moved. And then we also see oil from, which is actually heavy oil, from Prudhoe Bay at 5 degrees, from a recent paper by Minosa and Holder. And they were actually getting similar results in terms of degradation rates in a matter of a few days, as was some studies done in the St. Lawrence Seaway and a study in China. And we see quite a bit of agreement here. Now, I added this in here just to tell you that when the oil gets into the sediment, it's a different story. So there's a lack of nutrient diffusion and things like that in there. It goes anaerobic. And so that oil that got to the shoreline is going to stay there for a while, and it's going to be found in the depth for quite a long time. Now, in Science Express in January 6, Valentine and Kessler showed that, in fact, the methane went down very rapidly. Initially, we didn't see much degradation of the methane. That was a little bit puzzling to us, but I'll explain that in a minute. But after we got into late July and early August, the methane went down dramatically. The amount of DO dips went up dramatically. And of course, they also linked this to some clone libraries that they had and some direct analysis for methane monooxygenase and things like this. We also have recently shown that when we do look at the metagenome and metatranscriptome, that in fact, close to the wellhead, we see a dip in the metagenome, or I should say the metatranscriptome, in the amount of methane monooxygenase. So the amount of oxidizing, abilities to oxidize methane goes down quite a bit. But all the alkanes are quite a bit higher. And if we look at the proximal versus the uncontaminated, eventually when it gets out to the distal part of the plume, it actually goes up slightly higher than the uncontaminated, and we see that continuing. Now, there's a little bit too much data here, but we could spend a lot of time on this. So this is the metabolome, and this is IPATH, and this is just showing some of the metabolic functions. The red here is both what we saw in terms of single-cell sequencing, and then the metatranscriptome also agrees with that. The blue is where we found only the metatranscriptome, and the yellow is only where we found the single-cell sequencing. Now, just to summarize that for you all. So the Oceanosporiles possesses genes that code for chemotaxis and motility, as well as those that code for the degradation of C6 through C20 alkanes and cycloalkanes. Monooxygenase decreased probably due to the fact that the hydrocarbons, these small hydrocarbons and these alkanes are much better food. You're not going to go to methane unless you have to. And in fact, that's what we found, and also Kessler and Valentine found, that once the oil was gone, the methane degraded quite fast. The Oceanosporiles also exhibited a very rapid chemotaxis response, and we saw this in terms of the metatranscriptome, to the presence of oil and aliphatic hydrocarbons, but not to methane, which is not, not so, those weren't so abundant. Now, we had a number of important reasons that we think this might have had even a better effect on the Gulf, because it happens that I have a patent on a process for injecting methane underground. DOE owns it, and it's been licensed by 30, 40 companies. It's been used all over the world for the last 20 years. So we inject methane, low concentrations, into groundwater, and it degrades oil and trichloroethylene, because the methanotrophs will degrade 300 other compounds fortuitously. Now, some people say co-metabolically. I prefer to say fortuitously. So we may have gotten a deep cleaning effect after all of the oil disappeared and then the methane went down. So even some of these harder to degrade compounds went away. Now, where's all the oil? We took 170 samples from July 27th to August 26th, from 5 kilometers to 310 kilometers, and we were looking for that plume, and we could not find it. We went out and did transects trying to find that plume, and we couldn't find it. We went to the same depths. We knew that it was at, should be at that, and it was before, not there. The fluorescence would detect down to less than 1 part per million. And then we took samples back to the laboratory and tested all of those. And chemically, we could detect down to less than 2 parts per billion absent. So I don't say it wasn't there, but it wasn't, we couldn't detect it by any technique that we had. And of course, this is the time also that the methane started degrading quite dramatically from the Valentine and Kessler paper. Now, if I had to look for oil degrading bacteria, the Gulf of Mexico would have been the place. You know, it's 400,000 barrels of oil go into the Gulf of Mexico on average every year, and this is according to the National Academy of Sciences from their 2003 report on a 10-year average. And sometimes it's as much as 1 million barrels of oil a year. And that's been going on for millions of years. These bugs have to be adapted to the oil, and I think that's what we're seeing. Now, there's more natural seeps in the Gulf of Mexico than anywhere else in North America. The only place that is even a rival in any way, shape, or form is Santa Barbara, close to us. But notice up here in Alaska, where the Exxon Valdez spill, no natural seeps, no adaptation, and the oil went out over the surface and killed 250,000 birds. These are some of the pictures that we will never forget. This picture has been downloaded over 8 million times from the web. As near as we can tell, we saw about 6,000 birds killed, and of course 11 men, which I don't think gets enough attention, because they come from an international community. It's not like coal miners where they're all living together, and I think it's a tragedy that we haven't given them more attention. We also saw potentially 90 marine mammals. But compare that to the Casco-Busan spill, we had 2,500 birds killed in the Casco-Busan, and that was minuscule, right out here in the bay. But at Exxon Valdez, 200,000 birds, 22 killer whales, 2,800 otters, and the list goes on and on and on. And we can still find oil in that rocky coastline down deep in a few places. But about 99% of it's been degraded. That was, again, an order of magnitude less. The Ixtoc-1 blowout down in the Gulf of Mexico, 3.1, but our best estimate is maybe about 10,000 birds. But look at what the Ixtoc-1, how it compares to the BP blowout. So Ixtoc-1 went on for 9 months. We had to bring in red Adair, and us older folks can remember red Adair. And look at the amount of coastline that was oiled, way up into Texas, and how far that plume spread. Again, it was 50 miles from shore, just like the BP. It wasn't as deep. It was only 170 feet, 70 feet deep. But it was the same type of oil. It was light crude. So that is more inherently biodegradable than the heavy crudes are. Now, Houston Chronicle is having fun at our expense. Huge oil-eating microbe just ate Earl. And I put our microbe down here, so I put that in. So if you have more questions about oil spills and microbes, I would suggest that you go to a recent report that a bunch of us got together at the American Academy of Microbiology and put together. Really some of the great experts in petroleum microbiology. And we put frequently asked questions together. You can download some of this on the web, and it will give you some of the answers that you were dying to know. I sure wish we would have had this when the oil spill happened, because maybe some of the frenzy would have been dissipated. Now, I don't want to tell you that this wasn't an ecological disaster. It was. But it wasn't as bad as some people portrayed it to be. And there will be a lot of studies that we need to do with the dispersant and the correxit. The dispersant does grade much more slowly. However, the dispersant also, a major component is propylene glycol in the dispersant. And just to give you a perspective on risk on that, some of us that are older have had a colonoscopy. The night before you have a colonoscopy, they give you propylene glycol. It is a really effective dispersant. I can tell you that by personal experience. And we would hope that our physicians haven't been given this to us for decades, that they know the health risks of it. So, you know, there's a lot of frenzy. There was a lot of frenzy about the oil spill. There's a lot of stuff that we still need to do. There's a lot of studies that we are still doing. But it certainly was an ecological disaster. But I think this helps explain, using the systems biology approach, explain exactly what was going on. Thank you.
Questions for Terry? Okay.
So one question might be...
Oh, Jim, you can't ask questions. You know all this.
that the organisms degrading the oil then would consume the nitrogen and phosphorus, which might deplete the photosynthetic population. Was the mass of oil in that region enough?
No.
Okay.
No. That's why I kept saying the concentrations were really low. And it was spread out and it was constantly mixing. So as I said, it was just slightly higher. The average was slightly higher than 1 part per million. That doesn't even break standards for most things, risk standards. Yeah, and so that explained why we never saw extreme depletions of these limiting nutrients and that sort of thing.
This is a minor issue, but this subspecies which have high expression of particular group of MCPs, when you compare similar species in other places, are they newly evolved, new gene, or it's just the expression level difference?
Well, they're not newly evolved, probably. They probably have been down there for a long time. This is probably one of the first times that we've been able to do this much sequencing, and especially any single cell sequencing from JGI that has ever been done at this depth. And we could directly go from picking the cells out and not have to worry about culturing them and picking them out and directly go to sequencing. The co-LEAC and a lot of others we have isolated, but the ocean Aspergilli strain we still haven't isolated. It's quite sensitive to, I think, to temperature. We do know that pressure is probably not an issue down deep here. And Scripps has done some studies on that. Yes, sir.
Looking at the speed of degradation of hydrocarbon and the temperature and the flock, all this putting together, do you see them accumulating intracellular compounds or extracellular polysaccharide that fast? That's how they are able to not degrade but hide the material very quickly? Meaning, if they're making some sort of a polymer and putting inside the cell like PHB, or they're making lots of carbohydrate, putting in the cell, outside the cell, making flock, that's one way they can take it up very quickly.
Yeah, we don't, so they are not, they're actually incorporating it into their cells. And we've never shown that in any oil degradation that there is any other component that's found in the cells that's toxic from the oil. So it's unlike metazoa, which will concentrate things in certain parts of the body and that sort of thing. We don't see that in the bacteria. And we actually talk about that in this document from the American Academy of Microbiology, too. Yes, sir?
Terry, this is sort of a follow-up to Jim's question, but about oxygen as a limiting nutrient and how, especially since that is poorly soluble, it might even have the same answer. So
yeah. So you have to remember the current was moving at 7.8 centimeters per second.
Yeah.
And so it was constantly mixing. And so that was replacing any oxygen, that depletion that we saw. We never saw the oxygen go down to low levels. And low levels, and that was one of the things that EPA was concerned with. And so we were measuring oxygen and using a variety of techniques for measuring oxygen. And we never saw it. We saw dips, and that's why I call it a dip. So we did see some significant drops in oxygen associated with the oil. We also showed that it was not the oil that was causing those dips chemically. It was actual, and it wasn't differences in the membranes and permeability and that sort of thing. We tested all that. The concentrations of oil were not high enough to cause any of those things.
And then I think this is beyond what you were looking at, but were there any strange interactions between the Gulf hypoxia zone and the high nitrate plume coming out from the Mississippi?
So as I said, it was going, so I have, if you go to YouTube, you will see the longer version of this. And I do talk about the dead zone. And the dead zone is up to the north west. It's much shallower. And we don't believe, and this is the government agencies and the OSAT report, which is Operational Science Advisory Team. We don't believe that it had any effect on the dead zone. It was an unusually hot summer. There was a lot of rain this summer, and a lot of things got washed into the Gulf. There was a slight increase in the dead zone, but we don't think that that's directly attributable to the oil. Another thing that happened is that the fisheries were closed for several months. NOAA has tested those shrimp and fish to the nth degree, and they cannot find anything, Crexit or oil. And yet, when they opened up the fisheries, they were getting larger catches than they ever got before, higher than ever recorded, but they can't sell it because consumer confidence is so low, especially in the big cities like New York and Chicago and DC, that restaurants won't buy it. So they're freezing it. So it's interesting sociologically, where are we getting seafood in these restaurants now in the big cities? We're getting it from Southeast Asia, which is hardly tested at all. Does that give you more confidence? It's going to take us a couple of years to rebuild that confidence, consumer confidence in Gulf seafood.
Say if you had a region where you had a spill, but there weren't seeps, would you ever think about pumping these microbes there rather than the dispersants?
I normally don't believe in bioaugmentation for oil because normally within 72 hours, we see an increase in oil biodegraders. So I don't normally believe in bioaugmentation at all, and especially this shows that, in fact, you know, the best thing to do was to skim that oil, get as much of that neat material up as fast as we could, keep it from going to shore. The dispersants, it looks like they helped. Of course, there's a lot of controversy about that, but it looks like they did help, and it looks like it kept some of the material from going to shore. We did not use anything but natural attenuation on this oil spill so far. And there is calls for digging up some of the coastline, and some of the marshes and things like that. You know, that usually causes more long-term problems ecologically than leaving the oil alone, where it would be permanently interred and really represents a very low risk once it gets deeper and deeper, except if there's a hurricane and it plows it all up.
Wasn't your data where you showed less damage to wildlife in places where there weren't seeps? Wasn't the logic there that you didn't have the microbial communities there? Or was that your
Well, that could happen, but you know, all oil spills, like all politics, are local. You know, it's just, you know, it's a combination of when it occurred, the depth, the type of oil it was, if there was natural seeps there, the amount of mixing, storms. You know, 2 days after the Exxon Valdez spill, there was a major storm of 50-mile-an-hour winds. They couldn't use dispersant because of that. They wanted to. It blew everything to shore, and so it oiled tremendous amounts of coastline, and that's probably what caused the major amount of damage there. And they did use a lot of bioremediation strategies there. Now, Ron Atlas and I have a paper that we're preparing for ES&T, an invited paper comparing the Deepwater Horizon with the Exxon Valdez. And I can tell you now there's very little comparison. There's a lot of contrast, but not much comparison.
Great. Well, thank you very much. I want to thank both plenary speakers.